quite a tale. A mouse has been hiding its armor all this time. Researchers just discovered that the spiny mouse was concealing bony plates beneath the skin over its tail. At first it looks like a slightly more hairy rodent, but the spiny mouse's body is full of secrets. Found in rock outcrops throughout Africa and Europe, its back is full of porcupine-like quills made of stiffened fur. It has soft, easily torn skin and a remarkable ability to regenerate, like a species of desert gecko. Now, researchers have revealed another surprise in the journal iScience on Wednesday. Their tails are lined with osteoderms, or bony plates, making them only the second group of living mammals known to be equipped with underskin armor like an armadillo. Although spiny mice are widely known and commonly used in all sorts of lab experiments, nobody had ever noticed they had these, said Edward Stanley, a biologist at the Florida Museum of Natural History and an author on the study. The discovery came when he was CT scanning specimens for the Open Vertebrate Project, an effort to build a public online database of 20,000 vertebrate specimens from museum collections across the United States. X-rays of the mouse's tail gave him pause. They reminded him of the lizards he had worked on for his PhD. But the only living mammals with known osteoderms were armadillos. I know enough about osteoderms that it's a fairly unknown thing for rodents to have them, Dr. Stanley said. The discovery was serendipitous, said Malcolm Maiden, a biologist with the University of Florida and an author on the study. Dr. Maiden already had a long-standing research project built around spiny mice, centered around their remarkable ability to regenerate skin, muscle, nerves and parts of their spinal cord. The researchers joined forces, studying how the osteoderms developed over a mouse's lifespan and sequencing the species RNA in an attempt to identify the genetic switch as responsible for the bone armor's development. Dr. Stanley also scanned specimens of the spiny mouse's closest relatives, the linkrat, brush-furred mouse and Rudd's mouse. He found that all three also had armored tails, while more distant relatives did not. The discovery suggested that a common ancestor of all four species possessed the trait. The purpose of the osteoderms isn't clear. Spiny mice may use them to shield themselves from predators while burrowed in crevices, Dr. Stanley said. Another possibility. While the mice's skin tears easily, the armor might help protect the inner tail structure, like wearing chainmail under an easily removed glove. Osteoderms have re-evolved at least 19 times in different lineages of animals, Dr. Maiden said. They are often found in reptiles such as lizards, crocodiles and non-bird dinosaurs. They have also been found in a few extinct mammal groups like immense armadillo relatives called glyptons and giant ground sloths, whose skin armor the spiny mouse's closely resembles. Finding osteoderms in a fast-breeding, easily maintained animal like a mouse could help unlock how and why the forces of evolution have continually produced underskin bone armor, Dr. Maiden said. Now that they have narrowed down a list of genes that might be responsible for this trait, they can try to produce osteoderms in lab studies. I want to work out what genes are responsible for making osteoderms and then make a lab mouse with armor plating, Dr. Maiden said. The building blocks for osteoderms might be in the heads of vertebrates, Dr. Stanley said. The vertebrate skeleton is largely formed of cartilage that grows bonier over time, but the skull bones form through hardening collagen, which the team suggests might have been repurposed from the armored heads in early lineages of fish. If you can grow a skull, you have the genetic architecture to grow bones in your skin, Dr. Stanley said. The trick will be to use genomics to figure out whether the mice's tail osteoderms form like their skulls. That would lend credence to the idea that osteoderms went from armor to skulls back to armor. It's also possible that osteoderms, which are generally tucked discreetly under fur and skin, may be considerably more common in mammals than generally thought. Nobody has actively gone looking for them, Dr. Stanley said. It took exploratory science like the Open Vertebrate Project to find them, he noted. Dr. Stanley hopes data from the project will lead to similar discoveries. Building that kind of accessibility to museum samples and the digital data pulled from them will have benefits for all kinds of fields, Dr. Stanley said. After all, we didn't know what we were about to find. Newly discovered gecko escapes danger naked and alive. The fish scale gecko has a freaky way of eluding danger. When snatched by an attacker, it rips off its scales and skin so it can slip away unscathed. Basically, it streaks to survive. It looks like a fish until you grab it, and then it looks like a naked chicken breast, said Mark D. Schurz, a doctoral candidate at Ludwig Maximilian University of Munich. The torn away scales reveal the gecko's pink flesh, and through its translucent tissue you can see its spine and blood vessels. It's bizarre, it's really surprising, and it's quite uncomfortable when you see them, he said. It may seem like a gruesome getaway, but it doesn't hurt the lizard. It loses its skin and scales with extreme ease and regenerates them in full a few weeks later. The new scales grow in with a different pattern than the previous ones, but other than that are nearly indistinguishable from the originals. Mr. Schurz and his colleagues discovered a new species of fish scale gecko in the limestone karst of northern Madagascar, and published their findings Tuesday in the journal PRJ. Known as Gecolpus megalpus, it has the largest scales of any fish scale gecko, some of which measure about 8% of its body length. That would be like a person having scales about the size of hands covering parts of the body, according to Mr. Schurz. You would think they are somehow protective, but really they are a decoy, therefore blocking the teeth or claws of whatever wants to eat them, Mr. Schurz said of the giant scales. They get stuck in those jaws or claws and allow the gecko to escape naked and alive. Fish scale geckos have long mesmerized herpetologists with their hooden-esque escapes, but scientists are still not sure how many species of the lizards exist. A genetic analysis in 2013 suggested there might be about 10 species, but, including the new discovery, only 5 have been recognized so far. Researchers are working to confirm the new species through different methods. Mr. Schurz and his colleagues ascertained that G. megalpus was its own species by using a micro-CT scan to compare its skeletal structure with the previously recognized species of Gecolpus. The subtle differences they found in its eye bones and other parts of its body, in addition to the enormous size of its scales, led them to determine that it was distinct, and the first new Gecolpus to be discovered in about 75 years. Though Mr. Schurz has never grabbed G. megalpus in the wild, he said he had had plenty of experiences trying to snag their kin. Because of their speed, agility and throwaway scales, they are nightmares to catch. Summary. Osteoderms are bony plates found in the skin of vertebrates, mostly commonly in reptiles where they have evolved independently multiple times, suggesting the presence of a gene regulatory network that is readily activated and inactivated. They are absent in birds and mammals except for the armadillo. However, we have discovered that in one subfamily of rodents, the Diomean, there are osteoderms in the skin of their tails. Osteoderm development begins in the proximal tail skin and is complete 6 weeks after birth. RNA sequencing has identified the gene networks involved in their differentiation. There is a widespread downregulation of keratin genes and an upregulation of osteoblast genes and a finely balanced expression of signaling pathways as the osteoderms differentiate. Future comparisons with reptilian osteoderms may allow us to understand how these structures have evolved and why they are so rare in mammals. Graphical abstract. Introduction. The bones in the vertebrate skeleton develop from 2 independently derived pathways. The majority of the postcranial skeleton and chondrocranium is of endochondral origin, where a cartilaginous model is first formed which then mineralizes, while the bones of the dermatocranium, the skull roof, jaw, and clavicles develop intramembraneously by direct ossification within a matrix of collagen. The origins of these 2 pathways can be traced back to the earliest Eolus fish. Ostracoderms and their jawed descendants, placoderms, were covered in bony plates of dermal or integumentary bone, which represented the dominant bony structures of these organisms and are considered to be the evolutionary precursor to the bones of the dermatocranium. 1. The endoskeleton of these early vertebrates was entirely cartilaginous, mineralizing in osteichthyes and forming the endochondral skeleton. The relative proportions of these two bone-forming systems reversed early in the evolutionary history of bony vertebrates, with the reduction of the dermal skeleton and the endoskeleton mineralizing and taking on a more dominant role to permit a greater degree of movement. Nevertheless, the dermal skeleton has been retained in the bones of the vertebrate dermatocranium and the skulls of modern-day fish, which have a plate of dermal bone, much reduced in size from the ancestral form, but derived from a common ancestral integumentary element. 3 non-cranial dermal bones are retained in tetrapods in the form of osteoderms, clavicles, and the furcula. 4 osteoderms occur in numerous lineages of extinct reptiles and amphibians and are c.